to have Nathan and Christian Rutz here to talk about a number of overlapping between movement ecology and disease ecology, and opportunities for us to really push both fields forward by collaborating more. And I wanted to give a special welcome to Meredith Van Acker and also James Hassel. And I don't know if Suzanne is on here, but our partner at Global Health Program at Smithsonian, they're helping us to put this meeting together, and we'll also be helping to coordinate the Q&A afterwards. So our meeting's going to run pretty much as normal, but we have saved a longer time than usual for questions, answers, discussion, for everyone to. The goal is really for this to be a community. Everyone to come with an open mind, an opportunity to learn more about how you might integrate either one into your respective disciplines, and just in general use this as an opportunity to kind of take a break from maybe what you are normally doing, and think about new cutting edge ideas as a group. So let's see, we're going to start out with just some brief announcements, and we have and then we'll have two 15-minute presentations, and then the rest of the time will be reserved for discussions. So and just to note everyone, the meeting is being recorded. We will be pausing recording during certain sections because our presenters have agreed to share some preliminary results with the group. So we just ask everyone to respect that privacy, and we will make sure that the recording is acceptable to everyone before it goes out publicly. So any questions before we get started? Okay, and I would welcome you to use the chat or raise your hand if you have something to chime in as we go along. But yeah, usual Zoom rules. So, okay, we're gonna start out. Let's see, Jessie, are you on yet?
I am. I'm
okay,
here.
Great. We'll have you go ahead and give an update on the Landscape U program.
Great. So some of you may be familiar with the Landscape U program. I just put a link in the chat if you are not. But in short, it is an NSF-funded program through Penn State that's designed to foster interdisciplinary and applied conservation research, specifically at the food, water, energy nexus. And the Working Land and Seascapes Network, chiefly through Molly Dodge and Steve Canty, have been fostering a relationship, building this relationship with Landscape U over the past few years. And we've hosted 3 students so far as part of this program. They have funds for the next 2 years to support graduate PhD level interns working on 6-week projects. And so the goal for the internship for the students is to work in an area other than their thesis focus and apply the new skills that they've gained as they go through their PhD. Students have a broad range of knowledge, including social science, that could augment existing research, and they're available for both desktop work or field work. And I know 6 weeks seems like a really short time period, but the 3 students that we've worked with so far have stayed on and made significant contributions to advance some of the work in the WLS network, particularly with, they've worked a lot on mangrove projects, so specifically mapping and social science with mangroves. So Molly Dodge will be sending around an email in the near future. But if you're interested in proposing a concept potentially, please reach out to Molly or myself or Steve Canty, and we'll follow up to provide examples of previous concepts that have been submitted or to help you craft an idea. And that's about it. Thanks, Lucy.
Thanks so much, Jessie. Okay, we have Matt. Do you want to kick us off with center updates?
Yes, I'd be happy to. I have a couple of quick updates. The first is about a new paper from colleagues that we've worked with as part of Movement of Life that's on bull shark juvenile habitat use and movement ecology in the Indian River Lagoon in Florida. And the lead author is Michelle Edwards, who is a Movement of Life intern with us at CERC 4 years ago, I guess. She did 2 separate internships with us. And then also Chuck Bangley, who is a former postdoc with us with Movement of Life, are involved, as well as colleagues at Florida Atlantic University and a few others. But one of the cool outcomes of this paper was that we were able to show a much greater spatial use of the Indian River Lagoon as juvenile bull shark habitat. So we're hopeful that this paper will help lead to an expansion of the essential fish habitat designation for juvenile sharks in the area. The second highlight is a new proposal that was funded through the Undersecretary for Science's Life on a Sustainable Planet Pathfinder program that's going to combine tracking of alewife using acoustic telemetry in Chesapeake Bay during the spawning migration, but then combining that with some bioacoustics work looking at spawning splashes and also high resolution stream temperature network to really try to line up how individual fish are moving on and off spawning grounds when they're actually spawning and how that relates to temperature, and then trying to link that in to potential changes due to climate change and land use. So we're excited to kick that project off a little bit later, I guess early next year when the fish come back in January or February, because these are anadromous fish that are out playing in the ocean right now, but they'll show back up in Virginia spawning streams in early, late winter, early spring.
Thank you, Matt. And there's a link to the new paper in the agenda. Anyone wants to check it out. Autumn Lynn, are you on here?
I'm on. Hi, everyone.
Yay, okay. You want to give Migratory Bird Center updates?
Sure. Lacey asked for updates on our field work, our tag deployments this summer, and we have a lot. So the Migratory Bird Center deployed at least 300 tags on 13 different species of birds. 4 of these species were ones that we had never studied before. And some of these species are range-wide projects that were new this year. Amy Scarpignato is leading a range-wide project to study Western meadowlarks and Eastern meadowlarks across the continent. We have a few shorebird projects on short-billed dowitcher and stilt sandpiper, a black tern project, 3 Jaeger species. That was my work, and this was the final field season for our connectivity study, but we might start an accelerometry study next year. The Jaegers are really cool species, and they're already in the Southern Hemisphere after leaving Alaska about 2 weeks ago. Nate Cooper led a new project on Merlin in the Great Lakes, and he is working on a translocation experiment to see if translocating Merlin will affect predation on piping plover. So a pretty cool study. And Andy Boyce is studying Sprague's pipit and chestnut collared longspur in Montana. So over 300 tags and 13 different species for us this summer. Most of those were from the Migratory Connectivity Project work. We have a fellow that worked with us this summer. I saw Dennis on the call, and now he's going to work with a new Movement of Life team for another fellowship. But I just want to thank Dennis Kim, who did a great summer fellowship project looking at wind effects on Arctic tern movements from our Migratory Connectivity Arctic tern study in both oceans, the Atlantic and the Pacific. So thank you, Dennis. And Jared, you're lucky to have Dennis working with you for this fall. We have a new fellow who is starting soon, Lisa Kettemer. She is from the University of Norway, and she is going to be doing a humpback whale connectivity study with me. So she'll be starting in a couple of weeks. And a final update, the Smithsonian and 2 other editors have finally signed a contract for a new book called Conserving Migratory Species that will be starting on this fall. So I'm one of the editors for that. Ed Gallo and Rich Fuller are also the editors. So I know a number of you on the call. Hopefully we'll be inviting folks to think about contributions to that book. And it's meant to be very cross biome, cross taxa. So lessons for different types of migratory species from different biomes. So really joining lessons across taxa and biomes into joint chapters rather than separating everything out by taxa. So we should have fun with that over the next year. So thank you, everyone.
Is that all that you did this summer, Autumn Lynn?
The month of fieldwork was good for my soul.
Pretty impressive. Okay, thank you so much. And Jared, you want to give updates from Conservation Ecology Center and other SCPI programs?
Yeah, sure. I guess the exciting thing and thing that I'm really happy to report on is we're seem to be getting back to normal with some of our field campaigns. And so in the next few months, we're going to be pushing on some collaring activities. Catherine Mertes just got back from collaring addax and scimitar horned oryx in Chad. And next month, I'll be traveling to Kenya and then Morocco first to tag Maasai giraffe with the Giraffe Conservation Foundation team. And fingers crossed, potentially wildebeest if we can get the permits. And then on to Morocco for a reintroduction of addax. And I know that Meredith Van Acker is also working for in Kenya to be placing tags on a disease study in Nairobi as well. So lots of activities, promising activities in the next few months. We had a paper that was recently accepted in Ecology and Evolution on the movements of elephants in Namibia. That's led by a former SCPI intern, Lorena Benitez. So it's not out in print yet, but certainly probably in the next month. And also very happy to report that the Iridium agreement that we've been working on, which will provide some funding for tags, but it also provides support for the communication charges. That was recently signed. So we're now moving forward, and right now that will provide tags for giraffe, for jaguar, and for a bobcat study here in Virginia. And then the last thing I just want to acknowledge was the large grant that Gila Shimon and Bill McShay and colleagues received from the Paul Allen Institute. They received 3 years of funding and roughly $1.5 million to study the reintroduction of black-footed ferrets in Montana. So another really exciting opportunity for this team and for activities in Montana.
Absolutely. Thank you, Jared. Okay, so, oh, and then do you want to speak briefly about the opportunity with the University of Glasgow for Masters of Science pairings?
Yeah, sure. So I think, over the past couple of years, we've had a really strong collaboration and growing partnership with the faculty at the University of Glasgow. And, one of the things we've always talked about is we have a wide diversity of data sets here and a wide variety of expertise. And so with this program over the past few years, we've been able to supply data sets and more or less ideas on projects that we don't realistically have the time to work on. And that has meant pairing with faculty at Glasgow, investing in students to try to push a lot of these projects to the finish line. And so this past year, I worked on a project in the Masai Mara, working on connectivity with Imogen Schwander, who's a master's student. We are going to actually be helping support her as an intern, both between Glasgow and Smithsonian. But also, maybe some of the Glasgow team can talk a little bit more, but I believe that the ideas for these master's projects usually come out in September. So potentially within the next month, some of those pairings will start to happen again. So the point here is just to say, if you have data sets, please feel free to voice your opinion and your thoughts on what might make a really good master's project. And we'd love to have you talk with the Glasgow team and see if we can pair your data set, your ideas with the student.
Yeah, and wanted to plug Michael Butler Brown's collaboration that was very successful with Giraffe. So yeah, 2 students who focused their entire master's projects on Smithsonian data sets. So really great opportunities to also strengthen ties with the team at Glasgow, and see a few of you on here right now. And oh yeah, and Autumn Lynn too. Yeah, you did that the year before. I think you were one of the first pairings. So we'd love to keep growing that partnership. And if I think they're going to start collecting project ideas to shop around to students next September, so next month. So get in touch with me if you'd like to stay on that email communication chain. And Chris, I just realized that your Wildlife Society workshop didn't make it onto the agenda. Do you want to put a link to that into the chat?
Yeah, sure. I'll throw a link in there. So TWS in November, we're doing a full day workshop, which you can still sign up for if you're going to TWS. And if you have any problems getting in, I'll make sure the space is expanded so that you can get in. A bunch of home ranges and take the contours and merge those together in range. Code where there's actually but
Perfect, thanks. Okay, and just a brief update. Movement ecology events, any recorded workshops, anything you'd like to share with the ecology community, especially the Smithsonian. And movement ecology work, and then events happening and jobs also from the broader movement ecology world. So for anyone across the Smithsonian and our partners to be represented there, just send me an email with content. Okay, and then I want any other last minute additions to this announcement. Sorry. I just wanted to acknowledge one departing and one new staff member. So we're welcoming Nicole Golden into Brett Jesmer's lab at Virginia Tech. And Brett recently started a position there and been a great new collaborator of our group, focused on building out movement ecology research in Virginia. And that's that podcast study that Jared mentioned. So Brett's welcoming his first PhD student. She wasn't able to join the call today, but we're hopeful she'll be able to present in a future meeting. And we're looking at connectivity across central Virginia. Cindy Obeth, who I don't think is on here. Yeah, Cindy has been an amazing intern for the last 8 months, really working with Movement of Life on a number of outreach communication projects. She's also been learning developing her R skills and species distribution modeling skills in Google Earth Engine. She really led entire presence at the Folklife Festival, which was focused on conservation in the Mara region of Kenya. And wanted to thank her for all of her efforts, and they're going to continue to be a strong partner in our work as we move forward. And just wanted to acknowledge her help all these months. Okay, coming events, I think I'm going to leave you guys to look through them in the agenda yourselves, just so we can save some time. But I guess the most immediate one is the British Ecological Society Movement Ecology meeting, happening at University of Glasgow in about a week. As far as I know, it's already filled up, but if you're in the area, I think it's going to be a great opportunity, featuring many of our collaborators there. Okay, and then media, I will also leave you to check out in the agenda with a special note on the Folklife Festival highlights. There's a webinar on corridors and connectivity that we did with the Center for Large Landscape Conservation. And it was a really great conversation with multiple leaders in the connectivity world. So I encourage anyone focused on connectivity to check that out. There's a link in the agenda. Okay. With that, we're going to move on into our presentations for today, and we're going to have Christian Rutz kick us off with his presentation on tracking the anthropause. And professor at the university in the School of Biology. He's also the founding president of the International Biologging Society and the co-founder and chair of the COVID-19 Biologging Initiative, which uses animal tracking data collected before, during, and after lockdowns to investigate human-wildlife interactions. Christian, welcome. We're really excited to have you here. I saw your talk on the Wild Labs, I can't remember exactly what they're calling it, but the series where for movement, to advance movement ecology research and technology. And I got very excited about some of the ideas that you were talking about. And we wanted to think more about how your experience working on this real global kind of synthesis work and large global collaboration, how we might learn lessons for more collaborative work across our disciplines moving forward. I'll let you take it away.
Thanks so much, Lacey, for the kind introduction, and thank you everybody for the invitation. I'm really excited to be here with you today and to have a good brainstorm about the future of our field and discuss with Ren and everybody else on the call. As Lacey mentioned, I will present very briefly today the COVID-19 Biologging Initiative, which is a global consortium for studying human-wildlife interactions during the COVID-19 anthropause, so during hard lockdowns in 2020. And I would like to emphasize that this is a major effort, and I've put the names here of our steering committee and acknowledge all their hard work, and I'm sure you will recognize many familiar names there. Well, human-wildlife interactions take many different forms. On this image here, you see extracting for human consumption. There are other very destructive of interacting with wildlife. You see a bit of Amazonian rainforest logged here, and the patch on the right has become completely unsuitable for most species. But there are more subtle types of interaction that are not necessarily lethal. For example, several things may affect its this. First of all, the road, the presence of the road, which may act as an area, but also the road. A red fox and a of those pedestrians in the ground of humans in a decision-making behavior and so on. And it's these more subtle interactions I will be talking about today. Huge progress has been made over the last few years in terms of assessing the impact of humans on wildlife movement. You're all familiar with Marley Tucker's landmark paper in 2018 in Science, where he and colleagues reported that mammals move shorter distances in areas with high human footprint. And the movements of this groups of animals. But the problem, and that is that the different kinds of impacts are usually confounded, and that makes studying their relative contributions very difficult. So when we talk about the human footprint, that has different components. First of all, it's us humans, it's the presence of humans in the environment that affects wildlife movements. Think about that red fox, that urban fox I showed you. It's our vehicles, our cars and our ships and planes. Think about the moose. But it's also our landscape modification, infrastructure, the roads and buildings we put the landscape. And given that in most occur, it's near establish what their relative contributions are in terms of disturbance effects and so on. But in 2020, the most tragic of the beginning of the pandemic, humans, which at some point resulted in more than half of the world's human population sheltering at home. Traffic was also significantly reduced. But importantly, what remained virtually unchanged is those static landscape modifications. The buildings and the roads, they were still there. They didn't disappear. So this created a really valuable opportunity, I think, to study the mechanisms of human-wildlife interactions. And we've all seen photos like this, anecdotal reports of animals showing up in unusual places during the first hard lockdown in April, May 2020. But of course, we are not interested in anecdotes. We want to quantify these effects. So the idea was born to use biologging to measure wildlife responses to these lockdowns and to analyze animal tracking data that had been collected before, during, and after lockdowns. When we had this idea in early 2020, we did a quick check about data availability, and we ran with Sarah Davidson's help from the MoveBank team a quick query on the MoveBank database to see which studies were actively collecting data at the time. And this is the snapshot we obtained, and that looked very promising. So there was clearly plenty of tracking activity at the time. We also had a look at the release of the first Google mobility reports that indicated changes in human mobility. We've plotted here a percentage change in humans visiting parks. And what you see here is a lot of red, which means drastic reductions in human mobility, but also some blue. So in some areas, there were actually increases in humans visiting parks. And it's these different kinds of responses that provided the perfect opportunity to examine how animals respond to these kinds of changes in human mobility. So the idea was born to launch a collaborative consortium. It was Fran Cagnacci, Matthias Loretto, and myself who started with early discussions. We then learned that other teams had similar ideas. And within really a few weeks, we formed a core group, which later became the steering committee of the COVID-19 biologging initiative. This group of organizations very quickly decided to huddle under the umbrella of the International Biologging Society. So the COVID-19 Biologging Initiative is now a working group of the society. We were delighted to receive financial backing from the Gordon and Betty Moore Foundation and National Geographic Society early on, which allowed us to recruit postdocs to push forward with data collation and analysis. And more recently, we've been endorsed by the UN Ocean Decade. But most importantly, when we put out a call for action and invited the international biologging community to collaborate with us on this big idea, we got a phenomenal response. By the latest count, we've got over 1 billion GPS fixes in our central database. This is data sets contributed by over 600 collaborators for approximately 13,000 tagged animals across 200 different species. We set up a portfolio of coordinated but self-contained projects. So our collaboration model was to, rather than concentrate research opportunities for just a few research groups or individuals, we opened this up and people could pitch their project ideas and launch projects that would then collaborate use this community resource of data. And we now have about 10 sub-projects. I will give a very brief overview of 8 of them. So we've got one that looks specifically at marine and aquatic animals, led by David Sims and Mark Handel and Alex Loveridge. We've got one on terrestrial mammals with global coverage that's led by Marley Tucker and Thomas Müller. And Lacey mentioned that a first publication output is currently under review. We've got a project led by Frank Cagnacci and Federico Ossi and Benjamin Robera on mammal activity and road crossing behavior, again, terrestrial mammals. We have one that has a geographic focus on North America and which covers both mammals and birds. It's led by a team based at Yale. We've got a group that looks at birds specifically with a taxonomic focus initially on raptors, with a separate project on gulls, and the idea is to use those as a springboard to looking at birds, at all birds globally. We are currently planning a new sub-project that looks at the weekend effect and how that may have changed during lockdowns. This is work that is in preparation. If you're interested in that, please let me know. We have a sub-project that looks specifically at issues relating to measuring human mobility rather than animal movements. It's led by Diego Elisoto and Ruth Oliver. And we've got a project that tries to characterize this unprecedented period of planetary calm, which we have called the anthropause, using satellite-sensed nightlight data. And that is in collaboration with NASA. And we are excited about that as well. And we've got other projects under development. Some of these sub-projects are still accepting data contributions. Others have closed their databases and are currently writing up results for publication. I want to, just as a very brief snapshot, tell you a little bit about the Raptor project and share some initial findings from that. We chose raptors for a variety of reasons. First of all, they are tracked quite frequently around the globe, so we expected that there would be good data availability. But also raptors as a taxonomic group have a multitude of complex interactions with humans. In some cases, they depend on food provided by humans, either deliberately or otherwise. In other parts of the world, they are persecuted by humans, which selects for shyness and avoidance behavior. So we felt. I just want to conclude with a few forward-looking comments, and hopefully they will be useful for sparking a discussion later on. So what are the advances we hope to produce with the COVID-19 biologging initiative? The big goal is to identify what makes winners and losers in the Anthropocene. I think this is the ambition of the overall initiative. And of course, at some point, we will pull the insights generated by these different sub-projects and look at the very big picture. We want to move from correlational patterns to causal relationships. We can do that to a certain degree because of this quasi-experimental intervention provided by lockdowns. That should allow us to develop predictive models of global human impacts on wildlife movement. And that in turn should allow us to design effective or more effective conservation interventions. But a really important output is that in the course of running this initiative, we've built a very inclusive, diverse, and collaborative community, which I hope will continue to work together well past the pandemic. A few lessons learned. First of all, not that I would have expected otherwise, but this community is just amazing. We were absolutely blown away by the generosity and support from the community when we put out the call for collaboration. And it's absolutely inspiring to see how people contribute their hard-won data to these joint analyses. And I should mention that, of course, data providers whose data are being used in our publications, they are offered co-authorship. At the same time, we learned that it's simply impossible to discover all suitable data sets. Of course, some teams decided not to contribute the data, but we continue corresponding with people who haven't heard of us despite our best efforts. And I've got an idea of how we can deal with that. I've got a paper coming out in a few weeks' time in Nature, where I suggest we need a global tag registry. That is not a data repository, it's a different idea. I would be happy to discuss that with you. We also learned that human mobility data products are usually inaccessible to us ecologists, or they are presented in a form that is unsuitable for linking with animal movement data. And that is something we really ought to address as a community. And the team led by Diego Elisoto and Ruth Oliver have a manuscript that focuses specifically on this challenge. We also found that our collaboration model worked extremely well. So this idea to have coordinated sub-projects that are led by different teams really worked in terms of empowering different teams to run with their own ideas, build their own groups of collaborators, and think of their own outputs. And of course, we've got the collaboration across sub-projects. I see significant scope for productive post-pandemic collaboration. So hopefully there's much more we can do than look at lockdown effects. And here are a few ideas for particular collaboration opportunities I see with the movement of life community. We've just scratched the surface in most of our sub-projects in terms of the movement analysis we do. We use very basic metrics. I showed to you median distance, hourly distance moved. Well, we can do much better than that. And you've got a lot of expertise in your team for taking that to the next level. We would love to discuss that with you. I mentioned this weekend effect sub-project. The idea is to see whether the weekend effect is being affected by lockdowns, so changes in human mobility. Really early days, would love to collaborate. We are thinking of mobilizing the community now to document effects of possible anthropulses. By that, I mean possible spikes in human mobility as we emerge from the pandemic. We really shouldn't miss that opportunity. We've documented the lockdown effects. Now we need to keep recording what happens as the world emerges from the pandemic. And the idea of this global tag registry, I hope you will support that. I've given that like a working title acronym, the Tag Registry for Advancing Conservation Knowledge Track. Would love to discuss with you how that might work. And with that, I will leave it here with a big thank you. Here's some contact details if you want to reach out. And I look forward to discussing and collaborating. Thank you very much.
Yeah, to introduce Ron, he is a professor at the Hebrew University of Jerusalem. I'm sure he's a very familiar name and face to many in this group. He's credited with really formalizing the framework for movement ecology as a discipline, and has been very involved in numerous global projects, and also a key thought leader in advancing the fields of movement and disease ecology together. So we're excited to hear about your talk today. Thank you, Ron.
Thank you very much, Lacey, and thank you, everybody. And thank you, Christine, for this very interesting talk and project. And so I've been involved with the Smithsonian more than 20 years, and I was actually affiliated member of the STRI in Panama, And so I like the Movement of Life project, and I look forward for exciting contributions from you all. What I would like to present today is a highlight on the current state of movement ecology and some future directions, with some emphasis on managing disease outbreaks. The thought was that if Christian covered the topic of COVID-19, then the world now face another global disease of avian flu. So these two can be combined under one umbrella, how animals and humans and viruses interact. So let me start with this, what we all know. This audience here all know that as the time now is ripe for big opportunities for movement ecology research. This framework that we proposed about 14 years ago has proven meanwhile to strongly integrate components and taxa and topics and movement phenomena, but we still need to do more. And especially we need to scale up from individuals to groups, population, and community. This is a call and some guidelines how to do this. And we still need to understand how movement scales up in time and scales. And there's lots of work, theoretical work awaiting here. Maybe a good opportunity to push this forward is this 6-month-long program in Cambridge, in the Isaac Newton Institute in UK. It is arranged by Luca Gugioli and colleagues, and there will be lots of opportunities, 3 workshops, 1 summer school, and really 6 months of opportunities to work together. And it's not only about movement ecology, it scales down to cell movement as well. And I invite you all to register for this program. The link is here. And but the majority of my talk will be on the tools, and especially the data collection tools and the data yield. This is a slide that I've been showing for more than 10 years. I was really amazed by this jump in orders of magnitude in the numbers of the data yield in my own group. And this is not only in my group, it has been highlighted in these 2 reviews published 7 years ago in Science on terrestrial and marine movements. So you see here the same rise in several order of magnitudes, and similarly it has been occurring in marine research. Early on, actually, I noticed that ecologists are not very reluctant to accommodate this revolution. Maybe because the ecology has a strong data poor history, and maybe because we do not have tools ready to analyze big data, and maybe for other reasons of being cost effective. What are you guys going to do with such a ridiculous amount of data? This was a fair note that Steve Swearer said very clearly in a presentation I gave in Scotland 9 years ago. And again, this is a fair question, and we work really hard to find good answers for these questions. And I think we do have some good answer for these questions, and they were published only now, after 9 years, in the beginning of this year, in this review in Science on high throughput movement ecology. localization attempts per dollar than compared to GPS based on satellites. The same principle is used is what is called acoustic telemetry in aquatic and marine ecosystems. It's the same equation, the same principles, and this has long history of more than 20 years. And in fact, reverse GPS was used in animal telemetry before GPS. The famous study of Craig brothers is actually reverse GPS and not GPS, because the calculation of the localization of this famous bear and famous elk, I think, was done actually by the system and not on the animal side. So another important feature of this system is that they are automatic, and this allows a jump of again 4 to 6 orders of magnitudes in data harvesting. So the idea to develop this system in terrestrial ecosystem was to find alternative to GPS that will work on smaller scales and will be more efficient and allow tracking smaller individuals and many of them simultaneously at high spatial accuracy and frequent sampling for long durations, and to get the price down by 2 orders of magnitude compared to GPS stars. And this sounds like a dream, but with a partner like Sivan Toledo from Tel Aviv University, he made it possible through this collaboration under the Minerva Center of Movement Ecology. We call this Project Atlas, and the first Atlas system was developed, implemented in the Hula Valley that I will talk on later on. And it's a flat valley surrounded by mountains. It's a key stopover site of the Palearctic migration system, and there are many cranes over there and many wildlife. So it's a good place to track wildlife. And meanwhile, it has been implemented also in the Netherlands, in UK, in Germany, in Canada in progress, in Mauritius, in Australia, and in Svalbard as well. And Hila Shamoun has discussed with us this ferret study that was mentioned earlier, and we hope there's a potential to establish the first Atlas system in the US with this collaboration as well. And so what are we actually gaining from getting 2.5 million localization per day? This is from the Atlas system in the Hula. We gain insight that we cannot actually obtain with other tracking devices, and these were described in these papers of the last years. First evidence for a cognitive map in wild animals and so on. And I would like to go through 4 major topics that can be advanced dramatically with the availability of high-resolution data. One of them, very general, is the issue of individual variation among individuals that we know is quite large in terms of behavioral traits and cognitive abilities and so on. So, for example, the shy birds here are less explorative than bold ones according to the high-resolution data. But if you subsample the data, you get very similar tracks, and this is 30 minutes intervals that considered by some movement ecologists as high resolution. I even read that 1 day or 1 week is considered high resolution by some standards. So the word high resolution is used is misused, I would say, and by several orders of magnitude. People say it's high resolution, so you really need to read the details and look at the sampling frequency. So we have different conclusion based on the same data sets and different resolutions. Another big group of questions is on biotic interactions between individuals of the same group, the same species, and other species. For example, what is the potential for disease transmission in this example? So there is high potential if you look at the high resolution data. There is no potential if you look at the subsample data. Different conclusion based on the same data set, but with different resolutions. Another group of question is on interactions with humans and the environment. And again, this hypothetical example shows that fish avoid vessels, whereas the subsample data, you get the opposite conclusion. And the last topic, general topic, is the one that I emphasized earlier, is the variation in space and time. And here, obviously, you got high resolution, then you can nail down the real local behavior. So this fish is actually searching what is called area restricted search in a scale of 100 meters. And this disappears if you subsample the data to 30 minutes intervals. So in these three main fields, we actually can progress much better than before with high resolution data, and it's coming. It's already available, and there are more and more. Also GPS and also computer vision is contributing to this, and some radars, and so on. And I think this will mark some of the landmark studies in the next years that we will see in this field. Now, moving to the more applied side of this revolution, and you probably all seen this highlight on the need for global monitoring, and it is supported by this group from Max Planck and in collaboration with the Biologging Society and so on. It's a wonderful global consortium that work on that. And also Modus is now providing quite large scale, continental scale, quite many stations, especially in North America. It does suffer from low resolution and low accuracy of the data. And now imagine if we have this network with Atlas stations that provide very accurate data every second. This I believe is possible. It sounds like an incredible dream, never not feasible. But we do believe it's possible, and I hope that when you invite me in a few years, I will prove to you that we can do it in a continental scale as well. But this is future, and what we can do now is to monitor, especially in real time, in the context of conservation, and we can look at, for examples, when lions approach farms in Africa, and these wonderful examples of albatrosses providing the means to locate illegal vessels in the vast oceans. So what I actually want to emphasize on in terms of opportunities here is, first, that this is a wonderful opportunity to make the observational movement ecology approach, which we all do, make it more experimental. This is a wonderful example of RFID-based experiment of wild birds and chickadees, but it does not contain any data on the movements, and we actually can do it with real-time tracking systems, and this is a potential that has not been accomplished so far, and we await to see the first studies on using this and being able to to manipulate specific animals in real time and conclude their behavior similarly to lab experiments that many people do for a long time. Data evolution can boost up experimental approach in movement ecology, can provide a cost-effective way to track movement across spatial scales for both basic and applied reasons, and steer the management of human-wildlife conflicts and diseases and so on. So I would like to thank my group mates and who were involved in this work, and collaborators, especially Sivan Toledo, for working with me on Atlas, and Nikki and Idan, who are here, that were working on the avian flu funding agency. And thank you very, very much for your attention.
Thanks, Ron. We actually lost Lacey. Looks like she's having some internet problems, but we have plenty of time here. We have about 40 minutes left on the schedule. So again, I want to thank both Christian and Ron for taking the time and sharing their research with us. I'd like to basically open this up first just for general comments and questions from the group, and then we have a few curated questions as well. So Cole, I see your hand is raised, so how about you go first?
I just thought that last talk was incredibly fascinating, and I sometimes don't listen to every meeting I'm in, and just the whole idea of, like when you were talking about disease detection, and then it reminded me of when we were tracking elephants in Myanmar and we found out that there was a poaching incident. I think coming up with these reasons, beyond understanding animal movement, and I haven't read that paper, but the whole, why it's important, like what we're going to do with such a ridiculous amount of data, saying, okay, these are the problems that we can solve or get ahead of through tracking. But the disease detection, I was just blown away by, but I think, poaching, and if there are other things, I'm a fundraiser, so that to make the case to raise money for this, it's important. So thank you. And thank you so much for a great presentation.
Thank you.
Yeah, Ron, just a quick question about the avian flu data and the cranes. My question is, how often do you get a chance to handle the cranes? Meaning, how often are you able to assess the flu over time?
Yeah, so this is a really big problem, and cranes are very difficult to trap. They are very sensitive. They have capture myopathy, and so very difficult to trap. And in fact, there's no much data on related to disease ecology. We've been working on this for last 7 years or so. We had the NSF funded project with Wayne Getz from Berkeley, and the title was Pathogens on the Move, but actually we were not able to nail down pathogens in previous years. This epidemic occurred after we finished the project. So we used the sampling that we have from feces and so on to advance microbiome research, which is not directly related to disease, but and Nikki is here also, and she was working on other projects here. Now with this crane, we actually have some failures with common technology of PCR, and some promising avenue. I can elaborate a bit more if needed, and but this is very preliminary stage.
Yeah, so Autumn Lynn, I see you have a question, hand raised.
Everyone, thanks for those great talks. First a comment and then a question. I really love the idea of TRACK, the Tech Registry for Advancing Conservation Knowledge. I think that's going to be exceptionally valuable. And a number of us on this meeting have been working to compile sort of a discovery process for tracking of birds of North America as a part of Audubon's Migratory Bird Initiative and our Migratory Connectivity Atlas. And as you mentioned, Christian, there are a lot of hidden studies that don't appear in the usual places. So it's been interesting to find out how many studies are published but not in repositories, how many are in repositories but not published, and so on. So just a +1 and big support for that idea. And then Ron, thank you so much for your talk. Working in seabird colonies this summer, I've encountered the avian flu, although in Alaska it's been less prevalent than some of the European and British colonies. But this week I received an email from the New Zealand Ministry of Biosecurity asking me for tracks of Jaegers as connection points to Alaska. And so there's a lot of concern about this. And I wonder if in your community tracking cranes or other species, have you encountered sensitivities to working with the birds during an outbreak, and or have you seen a need for perhaps global protocols? Should we be handling moving colony to colony during an outbreak? What are the considerations that you've encountered working in the midst of an outbreak, and how valuable is the information versus the trade-off of potentially being a vector yourself?
Yeah, so Nikki is in the audience here, and Nikki and I discussed this particular question this morning. And well, I would say that if you have previously tagged birds and you can use this information, it's the best case. You're not risking yourself. You provide lots of information, but actually we did engage with sampling the cranes. We were there in the middle of the crisis, in the hotspot of the crisis. And if I can show you guys, can I show again?
Yeah, for sure.
Okay. So,
let me know if there's anything you want me to pause recording for, if it's very preliminary.
No, we do not record this. Okay. You need to do modeling and proper analysis and so on, and so there should be, beyond the data, there should be task force of people that know what to do with this kind of information. And the second thing, I think that for everyday work beyond outbreaks and this kind of incidents, it could be extremely useful for movement ecologists to arrange their metadata. So every lab here is engaged, I guess, with lots of tracking. And as a PI, I can tell you that updating the metadata of all project is a burden, a big burden. And MoveBank is not sufficient to do it. And though it's a good platform and so on, but you still need to make sure that every tag is registered and updated and so on. So Christian, for the sake of all PIs here and elsewhere, this will be very, very important, I think.
Thanks, Fran. Yeah, I completely agree. And I was thinking that one way TRACK, the registry, could add value to the researchers is it could be the place where you have the metadata. As you say, it's difficult to organize in a consistent format, but also where you host in one place all the permit paperwork associated with a study. So that could be your area access license, etc. So you could move to a system where in future publications, you give a reference number for a TRACK entry, which includes your ethical approval, your metadata, your study permits, and so on. So I think there's a lot of scope for innovation. And just another comment on who should be doing the work. I think one of the key innovations here is that making an entry in TRACK would be much easier than uploading raw data. So you simply say, I put this tag on this animal, and then you will hopefully feel that you should share the data through a public data repository as well. But it's a very efficient way of letting the community know that you are working on something and that these tags exist on animals. And I just wanted to share one final point about TRAC, which I feel quite strongly about. Is I was thinking back about how we launched the COVID-19 biologging initiative, and we could do that as a team because the research groups that were involved in this had the resources they needed, the extra resources to invest into this. It wasn't initially a funded project. Not everybody in the community has these resources. So I think a registry like this would really make our community more inclusive, and that anybody who's got internet access and has access to email can run a query on the database and can start forming a consortium. You don't need access to major resources. You don't need privileged access to established research networks and so on. If you've got a good idea, that's all you need. And if you've got an idea on doing a big collaborative study on elephants, you check who's got elephant data, and you are in business. So I like the idea of this being a tool also for empowering groups that wouldn't otherwise be able to form these larger collaborative initiatives.
Yeah, well, I'm glad you guys are talking about this, and I'm also glad that Fran is here. He's obviously really important for these discussions on data standards and what a database should look like.
Yeah, just a single comment on the fact that this really goes hand in hand in what we have already maybe not fully obtained about data, movement data. So there are many initiatives and platforms, and for sure MoveBank being the big one, that really aim to share the data, but the final goal to have a fully machine readable standard for movement information is not there yet. And the tag is a big thing about it because it's the way we know about it. It's really an essential portion of the protocol that took us to know about what the animals are doing. So indeed, the conversation on how to identify the tags in a standardized way that would be then readable and findable is definitely a great starting point and the stepping stone for everything coming after, in my view. And so a bit in a similar way to what has been done for specimens in museums. The tag is really like, like that ID that allowed us to say, well, that animal is not one individual of unknown origin, but it's an animal that has been caught there, tagged, and for which we have a history after that. Yeah, and I agree that it's easier probably than accounting for tags, but Christian, do you think, and maybe Ran too, because we are living with such a huge data sets which really build upon lots and lots of tags, also thanks to reverse GPS. Do you think it's really doable to do this without first start, like a dedicated project that would invest on such a repository, for example? What is your idea about this?
For really big data sets, it's I would say a huge project. We're still not there even for Atlas. We work on that for 10 years. It's such a challenging task. So it's easy to say, it's not easy to implement, and but we are getting there, I hope. It's another way of thinking is needed because for GPS we were able to manage the data sets mostly with MoveBank. Once we started with Atlas, it's really something else. So we still work on this. It requires beyond awareness. It requires people who has knowledge in data science, and to hire these people is extremely expensive. And so there's practical reasons why this is still delayed, but I hope it will come up in the next months or years.
Yeah, Fran, I would say that like a tag registry cannot be launched without funding. I mean, we've both learned, like through work with the society on data standardizations, it's just beyond of what can be done in somebody's spare time. But I think a simple registry that just collects data, metadata for the tags and researcher contact information and the associated data security and administration would be a very cost-efficient method. And it would buy us as a community a bit of time to deal with the data archiving side of things, like Ran mentioned, like these massive data sets which need a lot more resource and expertise for proper archiving. And there are, in fact, precedents of where other communities have done something like this. So you may have heard of AviSample. It's a recently launched registry for biological samples collected from wild birds. So that could be feather samples for stable isotope analysis, or blood samples, or anything. And all the registry holds is meta information on the existence of the samples. It doesn't contain the genetic sequences or any downstream analysis results. And that is a fairly straightforward database to set up. And so, yes, it needs funding, but I think if there is the support from the community, I think we could, if we pull together, we can launch this fairly quickly.
Yeah. So I'm seeing that Lacey has rejoined us, so welcome back.
Thank you. Sorry, everyone.
We have about 10 minutes left to ask some more questions. One of the questions that I simply had was, you know, Ron, you talked about the microarray and some other methods for kind of merging movement ecology and disease ecology together. I'm also just wondering both your thoughts on how we actually tackle this problem of understanding disease and its impact on movements. And I wonder if, like, livestock provides information to us that is helpful. You know, I know, like, from a movement standpoint, a lot of the movement studies have started, especially collective movement. I know some of the stuff that Juan Morales has done, it's focused on sheep, you know, because it's systems that we can manipulate, and maybe livestock provides that opportunity where we actually can sample over time if the information that we're collecting actually is beneficial for the species that we're more, I guess, quote unquote, more interested in.
Yeah, I would say that wild animals are not very similar to livestock in the behavior, in the scale of movements, and many of the disease problems we encounter are operate at scales way beyond movement of livestock. And for birds, there's I don't know, 99.9% of the work on avian flu is on poultry. And I'm not sure that the insights for what happens with wild birds is really substantial. So I think what we need is a new way of thinking, is to enter these long traditions with new ideas, with another perspective. I feel resistance from this community. I feel that they don't want us to mess up everything. They have their own protocols. They had biosecurity is a holy grail, and this is what we want to continue. But still, we see that the problem is not resolved, we're getting worse, and they blame this external world of wild birds that bring the disease again and again, but actually to resolve it, you need to understand how and why and so on. So I would call for a big shift in the whole field, more toward wild animals, and I would allocate most of the funding if I was in a position to work on wild animals rather than livestock.
I do see value in setting up systems with easily accessible animals such as livestock, but I agree with Ran that we need to tackle the real challenge, which is studying these processes in wild animals. And one idea I think that is promising is that we may still be a few years away from actually tracking the transmission of real disease and real wild animal social networks. But we have made good progress as a community in terms of charting those social interactions. As you know, the literature on animal social networks is exploding. There are now technologies available for recording these encounters, proximity loggers and various other technologies. So we have good data, or increasingly good data on social encounters between animals, and that can be extended to include animal-human encounters. And what is possible is to use those empirical field-recorded social networks to run in silico simulations how a pathogen might diffuse in the real world. And so you can use the field recorded network and seed in a computer simulation a pathogen or something else, and see whether the connectivity in the network supports rapid spread. And that, I think, is something we can do right now with existing data sets. I think it's underexploited as a research approach. I see first studies coming out on that, but I think we can make good progress in terms of getting a feel for our systems before we get to the final big challenge of actually mapping disease in real time. I will disease transmission in real time. I will put a paper in the chat where we did something like this with New Caledonian crows, and we were not interested in the transmission of disease, but transmission of information. But we recorded the actual crow social networks and then ran computer simulations on those networks.
Great, thanks to you both. Any other last minute questions while we have Christian and Ron with us? Meredith, go ahead.
I was just thinking about kind of the role of biometrics in some of these more customizable tags. It sounds like maybe similar to what you're doing with Atlas, Rand, like LoRa units that might be able to, say, track animal temperature as it's monitoring locations and accelerometer data. And just curious if you foresee a future in these sorts of biometric tracking devices, and whether there's a role for that in understanding infection status or overall health of an animal.
Yeah, so I do see a future, maybe even more than just movement data, location data, than other sensors, may be more informative. What I actually show you is inference from acceleration data on the state of the animals. It's very, very first steps. There's much more you can do, and we know this field is really progressing very well. We know that now there are developments of onboard evaluations of behavior and states that can be reported in a cost-effective manner to the user. In addition to the location data, there's a group in Deakin that works on that and show this in, how's that called? Black Duck, I think. And I believe the potential is there, and there are people already working on that, and this will prove maybe with other sensors as well. Christian, what do you think? What other sensors can provide reliable information?
I agree. I mean, we need to think about the whole suite of sensors. And as you may know, I think Lacey mentioned that earlier, that Wild Labs is currently running a community consultation, a big horizon scan to think about new technologies, new breakthroughs, new sensors. Yeah, it might be worth talking to them as well.
Thank you both.
Yeah, maybe one more thing, if I may. I think that one of the main directions for future is to combine computer vision with tracking. And this is indeed being forward by artificial intelligence work and in exploding for other reasons like autonomic cars and so on. But we can benefit from this progress and get a very accurate, full information of what is going on in very local scales. And if we can translate this to what we see from the few individuals that are tracked, either with GPS or with sensors, then that could be very, very informative.
I think just one thought. I know that people will have to leave, but like I see huge value in the idea of having tags on animals that guide other instruments that are not on the animals to collect additional information on local context. And so, as you will know, there are pilot studies that have tags that guide drones for observing the environmental context around tagged animals, and I see huge scope for innovation there in that area. It's similar to the reverse GPS in that you don't have all the magic happening on the tag on the animal. You outsource that to instruments that are elsewhere. That could be satellites, but that could also be instruments in the habitat of the animals.
All right, I think we're at 11 o'clock here on the East Coast, so we're going to go ahead and wrap up, and with a big thank you to our speakers, Ron and Christian. I'm sorry I missed the earlier conversation, but I'm going to go back and watch the recording, and from what I caught, it was really informative and insightful. And we really want Movement of Life meetings to be a place where the broader tracking community can come and brainstorm ideas and really talk about these big ideas in movement ecology. And we appreciate you guys leading us in that effort. And we welcome you to come back to future meetings and recognize this is a long-term goal for the community. So thank you for sharing where you're at with us so far. And everyone else, it's great to see all of our partners. And our next meeting will be in late November. We haven't sent out any information on that yet, but you guys will hear soon. And we look forward to seeing you all in the future. Anything else, Jared or Meredith, before we go?
No, just want to thank both our speakers. Hopefully we get to see each other in person in the near future.
Yeah.
Thanks for having me. Really enjoyed the meeting, and I would love to continue the conversation and think about collaborations.
Great.
Same from here, guys. Thank you so much. Looking forward. Yeah.
Awesome. Yeah, hopefully the Gordon Research Conference might be the next place we'll see each other, if not sooner. All right, everyone, take care and have a good week.
Thanks a lot.
Bye, guys.
Bye-bye.
Bye.